Now let us study about the Archegonia of Markantia. As we studied in the last class, the Markantia undergoes sexual reproduction. For the sexual reproduction, it requires the gamete formation. The male gamete are found on the gametophore called as antheridiophore, and the female gamete is produced on a gametophore called as archegoniophore. And in last session we studied about the antheridiophore. Now we'll concentrate on the archegoniophore. Archegoniophore is a female reproductive organ which is found on the vegetative thallus body of the Markantia. They are produced on the midrib of the Markantia or in the apical notch. These are also grows erect or posterior to the land surface. It also has the stalk and the lobed structure. On the apex of the stalk there is a disc-like structure which is lobed. The number of lobes varies and usually there are 8 lobes present. In the case of Markantia polymorpha, they have 8 lobes in it. And coming on to the structure of the Markantia's archegoniophore, the archegoniophore of the Markantia is longer than that of the antheridiophore. The length of the archegoniophore stalk, it also has a stalk which is found on the dorsal surface of the thallophytic plant body, and these are elongated structures. They have the stalk and the disc-like structures. The stalk is usually longer than that of the antheridiophore. The length of the stalk is around 7 to 8 centimeters, whereas in the antheridiophore it was 1 to 3 centimeters. And other end of the stalk will be having the disc. At the apex of the other end of the stalk has the disc. On the disc develops the lobed structures on which the archegonias are produced. So how these lobed structures are produced or disc is produced on the stalk is by the dichotomous branching. As the thallophytes undergoes dichotomous branching, even the prostately developing vegetative structure also develops the dichotomous branching, and they forms the stalk. It undergoes 3 tiers of successive dichotomous branching to form 8 lobed structures. As it is given here. Actually, the thallophytic plant body grows erect to form a long stalk. On the long stalk, it usually the apical notch of the long stalk develops the lobe, the first lobe. The first lobe undergoes dichotomous branching to form 2 lobed structure. Okay, first dichotomous branching to form 2 lobed structure. The 2 lobed structure will undergo again a dichotomous branching to form 4 lobed structure. The 4 lobed structure again undergoes the dichotomous branching to form 8 lobed structure. So 2 lobe, 8, 4 lobes and 8 lobe. It forms 3 successive dichotomous branching to form 8 lobed structure, which is disc-like in structure, disc shaped. Usually the disc shape is flat, on which, okay, on the disc the archegonias are present. If this is the disc, on the disc the archegonias are present. So as that of the antheridiophores, the disc of the antheridiophores, the disc of the archegoniophore is also made up of is made up of epidermis. So when you take the VS, that is vertical section of the archegoniophore, when you take the VS of the archegoniophore and the section of the archegoniophore and observe it under the microscope, it appears to be like this. So here it shows it has a long stalk which has 2 regions, that is the region where the lower epidermis is present and region where the upper epidermis is present. This says it is the vegetative part of the thallophytic plant body, a gametophyte, where it has the ventral surface and the dorsal surface. The dorsal surface has the pores, that is the air pores, which helps in the exchange of gases, and it has the photosynthetic filaments and the air chambers, has that in the anatomy of the Marcantius thallophytic plant body. Even here it stores the same. So this confirms that the stalk, that is the archegoniophores or the antheridiophores, are the vegetative structure which is developed from the thallophytic plant body. Okay, so here when you consider the disc is flat, which is found on the upper surface at the apex of the archegoniophores, where the disc is made up of the epidermis, outermost tissue called as the epidermis, which is closely arranged and it is discontinuous in nature. It is discontinuous is because of the presence of the air pores. The air pores helps in the exchange of gases from the archegonia. And on the lower surface, just below the air pores, is the air chamber inside which the photosynthetic pigments are present. Okay, photosynthetic cells or filaments are present. And in between these archegonial air chambers are the archegonia which develops. Archegonias are developed onto the upper surface firstly, okay, to form a flask-like structure. Before fertilization, it is found on the upper surface of the archegonial disc, whereas as fertilization takes place and the archegoniophore gets matured, okay, it slowly bends, the upper surface or upper epidermis slowly bends towards the downwards. As it bends towards the downwards, the archegoniophore, which is upright flask-like structure which opens on the topmost position, and the ovule is present in the lower part. Okay, as it bends, it gets inverted where the opening becomes, okay, faces towards the downwards. And these archegonias are usually arranged in the acropetalous manner as that of the antheridia. Acropetalous manner means here the smaller archegonias will be towards the periphery. Smaller archegonias will be in the periphery, and the mature archegonias will be in the center usually. Okay, whereas as it matures and bends downwards, the position changes. That is, when it curves like this, in this way, the smaller archegonias at the apex will move towards the stalk, and the mature archegonias in the center will move towards the periphery. So actually the arrangement is through the acropetalous order, where mature ones are in the center and the smaller ones are at this apex. But due to the folding natures, okay, here the position changes and the smaller one becomes towards the stalk, and the larger mature ones towards the periphery. How this happens, we'll study with a diagram here. So this is the actual position of the archegonial disc. On archegonial disc before fertilization will be flat, and the archegonias are found upwards, facing upwards. The opening is towards the upwards, and the development of archegonia is towards upwards. As the fertilization occurs, the cells of the center of the archegoniophore starts dividing more due to the repeated divisions of the cell in the archegoniophores. What happens? The older cells will move and they bend towards the inward position of the archegoniophores. As it moves towards inward position, what happens here is the archegonias which was upwards will move towards the downwards due to the bending nature of the disc. As a result of this, you can find here the mature ones are in the center and the smaller ones are in the periphery. Okay, so as it moves towards the downwards, what happens? The smaller ones comes towards the stalk and the mature ones towards the end. So actually the arrangement is acropetalous, but as the maturation occurs, it is reversed, and upright position of archegonia, okay, becomes inverted where opening faces towards the downwards. Let us now study about the how the archegonia develops on the lobes of the archegonial disc. On the superficial cell, one of the superficial cells on the archegonial discs are distinguished itself from the other cell. How they are distinguished from the other cell? These cells becomes enlarged and highly granulated and then cytoplasm. These superficial cells which develops into archegonia are called as archegonial initial cell. These archegonial initial cells undergo transverse division to form two cells, that is the lower basal cell and outer cell. The lower basal cell are later converted into base, where the basal cell undergoes division and to form the stalk of the archegonia on which the archegonia are present. So they divide repeatedly to form a short stalk, whereas the outer cell becomes the archegonia. So this outer cell undergoes vertical division to form two cells called as peripheral cell. These two peripheral cell again undergoes a vertical division to form the axial cells, four cells. Okay, these four cells, when it is arranged like this. That is, three cells are found towards the periphery and one in the center. Has the arrangement, three cells in the periphery is called as the peripheral cell, and the cell in the center is called as primary central cell or the axial cell. The peripheral cell undergoes the vertical division. The three peripheral cell, when it undergoes vertical division, they form six cells. These six cells are called as jacket initial cells, which helps in the formation of protective jacket around the archegonia. So the primary in a peripheral cell undergoes vertical division to form six cells called as jacket initial cells. These jacket initial cells forms the jacket of the archegonia. Next, the central cell. The central cell undergoes division. Okay, so these jacket cells also, when they undergo division, the six cells undergoes transverse division. Okay, where these cells, six cells, undergoes two tiers of six cells, where upper six cells forms the neck of the archegonia, and the lower six cells forms the venter of the archegonia. The lower six cells bulges to form the venter, whereas upper cells, six cells forms the narrow cells to form the neck of the archegonia. And now coming on to the central cell or the axial cell. These axial cell, first they divide two. When they divide into two, they form the primary ventral cell and the neck canal cell. Okay, neck canal cell. These neck canal cell again divides to form four. They form a tube-like structure in which four cells are arranged. So in between the neck, so these are called as neck canal cells. And the primary ventral cell, again they undergo division. When they undergo division, the division is unequal. When it is unequal, one cell will become bigger and the other cell will be smaller. The smaller primary ventral cell is called as ventral canal cell, and the bigger cell is called as the egg cell. So egg cell will be larger than the ventral canal cell. On the ventral canal cell, they are the neck canal cells. So now, after the maturation, what happens? These archegonias are covered with the archegonial neck cells, which is at the tip, are converted into a cover cells. The tip cells of the archegonia are called as the cover cells. There are four cover cells which is present at the tip, which helps in the opening of the archegonia during the fertilization process. So this is how the archegonias are developed. So the mature archegonia appears to be flask shaped in nature. They have a bulged part called as the ventral part, and they have a narrow part called as the neck part, and at the end of the neck part there are four cover cells which helps in the opening of the archegonia during the fertilization process. And the neck part, inside the neck part, there are neck canal cells which is arranged in 6 rows. Each rows will have 8 cells, and towards the lower part they have the ventral canal cell and a large egg cell. So this is about the mature archegonia and how the archegonia develops. Thank you all. In the next session, we'll be studying about the fertilization process in the Marchantia.